Yo, listen up, I got a story about a teacher, Mr. Wright. Yeah, he's a biology creature. In the classroom, he's spitting knowledge like a preacher. With his lab coat on, he's the lyrical feature. He's got test tubes and beakers mixing up that potion, teaching us about cells and the process of mitosis.
Hi Year 12s, welcome to the last video for cells. This is additional to the other stuff that we have on the website, just doing a little updated video because we didn't cover this in the cell control PowerPoint. So looking at cell culturing in this video, as per usual, here is your need to know. Not much of a need to know, these are taken straight from the Stage 2 study guide. Don't need to write those down, but this is good just to tick off to make sure you've covered everything we need to cover. So, what exactly is cell culturing? Cell culturing is the process of growing cells outside of the body of a living thing. Generally, this is done in a lab setting. And we use cell culturing to grow cells and tissues from microorganisms, animals, and plants. This entire process is done through first dissection, so removing the tissue from the source organism using sterile equipment and chemical agents, and then growing that tissue that we've removed. So the separated cells are put into a suitable vessel with a nutrient medium, and they're grown in an incubator. This provides an artificial environment for those cells to grow. We include required factors such as oxygen for ATP production, nutrients for metabolism, growth factors such as hormones to stimulate growth and cell division, the correct amount of water to maintain osmotic balance, optimum pH, optimum temperature, and antibiotics to kill any microbes or bacteria that can get in there. There's plenty of applications for cell culturing, as we can see here. I'm not going to go through all of these. It's probably important that you remember 4 or 5 of these. You're not going to be tested specifically on these applications, but it is also always useful if you get a question such as give 2 applications of cell culturing or something like that. So knowing 4 to 5 of these applications, maybe some of the easier ones like cancer research or virology research or cosmetics research, or even food production, just knowing some of those in your head that they are applications of cell culturing can help you in test questions. As well as applications, there is limitations of cell culturing. So cell culturing could be contaminated by microbes, which is why we've talked about in the first one that we normally have antibiotics and things in there to prevent that from affecting the cell culturing. But if they do get contaminated by microbes, we can show false results. Generally, when we're doing cell culturing, we're inciting rapid growth, and when we have rapid growth, that incites mutations and genetic variations, which is always undesirable because it's going to change the cells that you're actually working with. Non-cancerous cells have a finite capacity for growth, and they'll stop dividing eventually. Usually that's around after 50 divisions, which means they have limits to prolonged use. Now, in class, we did talk about HeLa cells, and I have provided a video underneath these by TED Talks that does go through HeLa cells, and that might be worth watching as well. The last limitation that we will discuss is the fact that cell culturing can be expensive and time-consuming. Moving on, we're going to talk about some specific types of cell culturing, and we're going to look at some plants and some animals. So the first one we're going to talk about is micropropagation. So some plants reproduce through what's known as vegetative propagation. In this process, new plants are produced from existing plant structures, whether they be stems, shoots, buds, bulbs, as a specific part of a plant. The process involves we take that small piece of plant tissue, which we call explants, and we remove it through dissection or like pulling it off the plant, etc. That explant is then transferred into a growth medium with growth factors, which incite cell division, which means that the cells in that tissue or the explant will start to divide and form a shapeless mass, which is called a callus. That callus is then treated with hormones to induce further growth and the differentiation of cells, which eventually will lead to established plants. We then take those established plants, place them in soil, and grow them under optimal conditions, and essentially you have a direct clone of the plant that you started with before we did vegetative propagation. We also clone animals, and the best example of this is Dolly the sheep. There are other examples of dogs being cloned, or pet dogs and pet animals being cloned in China, which you can find on Google News. Might be a good idea for a she-task. We won't cover these in the PowerPoint, but something you can look up on your own. Talking about Dolly the sheep, so our first official clone, the process of cloning Dolly was, first off, a non-reproductive cell, a somatic cell, was taken from the udder of a donor female sheep, and it was cultured for 6 days. We then took a separate female of a different species of sheep, and we treated that sheep with fertility drugs to induce superovulation, which created unfertilized egg cells, which we could collect. Using a micropipette, we removed the nucleus of the unfertilized egg cells to create an enucleated cell, so an egg cell without a nucleus. And we then used electrical energy to transfer the nucleus of the somatic cell into the enucleated egg cell, creating a recombinant cell. That recombinant cell was then grown for several days until it formed an embryo, and the embryo was transferred to a donor mother that became pregnant and gave birth to the cloned organism.